Hey, welcome back. This will be the first lecture for anatomy and physiology for the spring semester. We're going to begin with chapter 14, the cardiovascular system, and we'll move on from there. I had a question about the picture last time. It looked like I was on a beach in Hawaii somewhere. That was fake. There was just an image behind me, and the light shining on my head was fluorescent lights, not the sun. Thought I'd mix it up a little bit here for you. I've been riding motorcycles since I was 16 years old. That's a long time. Anyway, I'll jazz it up with some different images. And eventually I'll do away with a picture of me so you don't have to look at my ugly face. Any rate, we're going to start with the cardiovascular system. And I want to remind you again, if you haven't had first semester, I'll point out some things that'll probably be good for you to review as we go along. Particularly the cardiovascular system when I get to generation of the electrical impulses in the heart. If you've not been exposed to the nervous system yet, spontaneous depolarizations, that'll probably seem a little far out to you, but I'll attempt to give you all the background you're going to need to understand that, okay? All right, I'll try to remind myself to hit this button and enlarge the screen. I forget to do that sometimes and leave the thumbnail sketches down along the side. So at any rate, the cardiovascular system, I'm going to start out really general with this. Today I want to cover primarily a little bit of physics of blood flow. And on the next lecture, we'll get into the electrical basis of the cardiac rhythm. In general, cardiovascular, the prefix and suffix, basically includes the heart and all the blood vessels. So it's a study collectively of the heart and the blood vessels. So again, we'll be emphasizing the heart a great deal in this first chapter. Kind of as a way to introduce this, the heart is a pump. It provides the driving pressure. That's an important statement because I'll talk about mean arterial pressure shortly. And we'll refer to that as the driving force moving the blood through the system of tubes, arteries and veins. The arteries distribute the blood from the heart to the various organs and tissues. The capillaries is where nutrients are released, waste products are picked up. Oxygen, for example, is released there and carbon dioxide is taken back up. And the veins collect all of this and return it back to the heart. So that's kind of a quick overview. And again, in this chapter, primarily blood flow today. Next time I'll get into the actual physiology of the heart itself. Okay. Some interesting facts. Somebody's actually sat down with a calculator and figured out that during a lifetime of about 75 years, both chambers of the heart pump about 400 million liters of blood. That's pretty astonishing. That's a lake, a kilometer long and 40 meters wide and 10 meters deep. That's a lot of blood. The cartoons that I'm going to use, of course, I'm going to use the ones from the text, but I'll supplement some of those with some other figures, as I've indicated to you before. This one that I've captured here, I've always found a useful cartoon that I pulled from Arthur Guyton's textbook of medical physiology. It's nice and clean. It describes the flow of blood from the atria to the ventricles and to the arteries and then back to the heart again through the veins. So you'll be seeing that again as we go along here, okay? All right, some basic functions of the heart. Let me see if I can move this. I've got things on my screen that you can't see. Delivers nutrients, oxygen, things like oxygen, metabolic substrates, carbs, proteins, fats. All the cells in the body ultimately depend upon the cardiovascular system to supply them with all of their needs. So nutrients is one need. Cells generate waste, as I've indicated, like carbon dioxide. They generate acids, all sorts of metabolic byproducts. These are taken away by the cardiovascular system. Cell-to-cell communication, things like hormones. brain hormones communicate with the gonads, for example. Gonadal hormones communicate with other target tissues like the uterus in the female, or the male vas deferens, the penis, and so forth. So there's communication going on, and all of that, the signaling molecules have to be transported. And to a very large extent, they're transported by the cardiovascular system. So the blood also contains things like growth factors, ions, enzymes, and various things. All of these are transported by the cardiovascular system. Defense against foreign invaders, antibodies, white blood cells, T cells, and so forth, cells that phagocytize foreign invaders. These get from place to place through the cardiovascular system. Blood coagulation, the blood contains the proteins that can coagulate if a vessel is ruptured or damaged and prevent blood loss. We can have inappropriate coagulation, of course, blood clots, which can be fatal, but we'll get to some of that a little bit later. But in fact, in looking at this list I put together here, there's almost nothing that I can think of that the cardiovascular system is not involved in mediating or carrying out for us. So it's a real appropriate place to begin our discussions this semester. Using this simple cartoon from the text 14.1, as I promised you I would do. And again, I'll be supplementing. I have some others that I'm anxious to show you. It's a useful cartoon to begin to get a lay of the land. The heart is going to generate this driving pressure, this force to move the blood through the system. The cardiovascular system is a closed system. It consists of a pump and then tubes that distribute the blood throughout the body. And then tubes, of course, veins, which bring the blood back to the heart. But it's a closed system. And I can use this as a way to begin a study of the cardiovascular system. And on the side here, again, as I promised you, I'll be typing little notes, comments, things to draw your attention to. So here I've said, as we develop this system, you ought to be able to trace the flow of blood through the system, both pulmonary and systemic, and name the various chambers and heart valves. Be a good place to start. So let's do that. For example, if I start on the right ventricle here, blood comes back to the heart from the body. The superior vena cava drains blood from the head, the arms. The inferior vena cava brings blood from the lower parts of the body, the abdomen, for example. It enters through the right atrium, flows passively through this AV valve, atrioventricular valve, and I'll detail all of that shortly, into the right ventricle. When the heart contracts, it ejects the blood into the pulmonary trunk. In this case, we're just showing the pulmonary artery here. There's a one-way valve, which I'll talk about shortly too, the semilunar valve. The blood then gets distributed to the lungs. And you'll notice that it's coming in blue, as the author says, to indicate that it's relatively deoxygenated. Now, it's not completely deoxygenated, but it's lower in oxygen, is all that means. So as the blood flows through the lungs, it gets rid of the carbon dioxide, picks up oxygen, and it changes to a red purplish color. And again, you can see this under a person's fingernails and where blood vessels are close to the surface of the body. The blood returns to the left side of the heart through the pulmonary veins. Veins always take blood back to the heart. Flows into the left atrial chamber, flows through an atrioventricular valve into the left ventricle. When the heart contracts, it ejects the blood through another semilunar valve and into the aorta. The aorta branches, an ascending system of arteries, take blood to for example, the head, the brain. A descending aorta takes blood to the lower part of the body, some of the thoracic cavity, the trunk, the abdomen, and so forth. So here's just a general, again, this is a generic type cartoon to show oxygenated blood coming in, giving up oxygen to the tissue, and leaving relatively deoxygenated and blue. Now there's some variations on a theme here, and I've copied this image over again to draw your attention to this. First of all, most capillary beds in bodies like the trunk, or for example, the arms, like the biceps up above, you have an artery coming in and it branches up branches out into a system of capillaries. That's what they're representing here, where you have capillary exchange. Oxygen comes out, CO2 comes in. And then it drains through a small vein called a venule, and then eventually into larger vessels that we call veins. You'll notice some variations down below, though. Blood passing to the GI tract, like for example, the intestines. When the blood leaves the intestines, it doesn't go directly back to the heart. The GI system, the blood first of all passes through the liver. And there, this drainage of blood, this venous drainage from the GI tract, mixes with fresh arterial blood to the liver. So you get an admixture of relatively deoxygenated and oxygenated blood coming into the liver. So you have your second system of capillaries within the liver itself. The first system of capillaries is in the digestive tract, for example, the small intestine or large, then venous drainage, and then another capillary bed. And then finally, these coalesce into venules and veins and take the blood back to the heart. This kind of a system we refer to as a portal system. This cartoon in our text illustrates two of these, one from the GI tract to the liver, and another one where there's a portal system. This is actually within the kidney. So you can see she has had the artist say kidneys and then bracket this. There's two portal systems. And again, when I get to the kidneys, I'll show you how that portal system functions when we discuss renal physiology. And then down below is just another, again, a generic cartoon to illustrate a classic artery, arterial, capillary, venule, and venous type system. These are two portal systems. We'll see another one when we look at the endocrine system. This involves communication between the lower part of the brain called the hypothalamus and the pituitary gland. So the hypothalamus, blood will drain from the hypothalamus into a system of like sinusoidal-like vessels, and then there'll be a second capillary bed within the pituitary gland itself. So there's three portal systems. The GI tract liver, the kidney, this is actually the nephron of the kidney, which we'll see later, and then the portal system that exists within the brain. So again, I've typed some additional notes here. The liver gets both a fresh arterial oxygenated blood and a relatively deoxygenated venous blood from the gastrointestinal tract. Some other things, again, when I type things on here, they're just to draw your attention to things that I think are important. You'll note that there are no valves. Note that valves are present in the heart and the veins, but not in the arteries. So again, if I look here at the aorta, the left ventricle is going to eject blood out of the heart into the aorta. Now there's a semilunar valve that guards the entrance into the artery. But as the blood flows through the aorta, all the way down through the systemic circulation, like through the trunk or the GI tract, there's no valves. You don't get any valves until the blood flows back into the right side of the heart. The first valve that's encountered there is the AV valve within the right side of the heart. So no valves within the arteries. Okay? Again, I'll detail the anatomy of the heart as we get deeper into the physiology of it. And of course, you're going to get exposed to a lot of that in laboratory, actually much more so than I will do here. I captured this other cartoon. It's a good example of me giving you a supplemental cartoon. It's not more information, but it's a picture to help you visualize where the blood is. The average adult male might have about 5 liters of blood in the body. Where is it? Well, this is a nice cartoon to illustrate where it is. The pulmonary circulation, the blood within both of our lungs, these are large organs, but at any one time, there might be about 9% of the blood that's in the pulmonary circulation. Now, that's going to vary depending on the extent of dilation and so forth of vessels, but roughly about 10% of that blood would be in the pulmonary circuit. The rest of it is in the systemic circulation. So I've got a pulmonary circulation, lungs, and a systemic circulation, which is just everything else. I can break this down a little more. The heart even though it's providing the driving force or driving pressure, that mean arterial pressure, only about 7% of the blood volume is in the heart. Most of the blood is in the venous side of the circulatory system, and especially the systemic circulation. So you can see, you can pick up almost any textbook, and they will tell you that about 60%, the numbers vary a little bit. This one says 64, but about 60% of the blood is in the veins. Why do you care about that? Well, if you were to infuse somebody with a half a liter of fluid, saline, you'll find out very shortly that the venous circulation is very compliant. And that's where most of that fluid would go, is to the venous circulation. It's also important to you in a clinical sense because when a person is given vasoconstrictors to bring blood pressure up, the veins function, as I've indicated down below here, as basically like a volume reservoir. And when they contract, they can move a lot of this blood, 60% of the blood volume, they can move a significant portion of that back into the systemic circuit. More blood for the lungs, more blood for the other organs in the body. So again, the information I'm giving you does have some practical importance, some clinical importance, and that's why I'm giving it to you. Okay, so this is a useful cartoon just to envision in your mind where the blood is. Okay, whoops, sorry about that. All right, we're going to start talking about blood flow. And one of the first things I want to do is to caution you about explaining physiological processes in teleological terms. So I'm saying in this image, avoid teleological explanations of physiological processes. There's no magic here. Everything can be explained in terms of chemistry and physics, and usually a combination of the two. But as an example, I'm saying blood does not, shoot, sorry about that. Let me back up again. I keep getting images popping up on the screen that I can't control. Blood does not flow so that the brain will receive adequate oxygenation. That's just silly. Now, teleologically, it's true, but we assign that benefit to it. We say that if the heart beats harder and faster, it'll pump more blood, and it'll pump more blood to the brain. And it does so because the brain needs more blood. Okay? The brain doesn't know it needs more blood, and neither does the heart. Heart rate increases, and blood flow to the brain can increase as a consequence of that. But it does so in response to different physiological mechanisms, which we'll get to this semester. For example, baroreceptor reflexes. If one is in a lying down position on the floor and they jump up vertically real quick, within a second or two, the heart will beat a little bit faster, the vessels will constrict, and blood pressure will go up. It's just normal physiological response. As a consequence of that, there will be increased blood flow to the brain, a little bit. But neither the heart nor the brain knows anything about that. Okay, so we want to strive to explain things in terms of chemistry and physics. So in other words, I'm looking for physical explanations of physiological processes. Another way that I might say that, for if I'm describing the kidneys or the lungs, I might say, what is the underlying physiological mechanism for increased heart rate or blood pressure or something in that nature, okay? This, again, cartoon 14.2, the graph, shows the mean systemic blood pressure starting in the aorta. Again, this is just a mean, or you can think of it as an average for now if you want, even though statistically it's a little different. You can see it decreases from the aorta into the arteries, the continued branching of arterioles, extensive branching in capillaries, okay? And then diminishes throughout the venules, veins, and vena cava that brings blood back to the heart. One of the reasons that it decreases like this is because the vessels branch. There's a single aorta, but it soon branches. There's carotid arteries that branch off and take blood up to the head, the neck, the brain. The aorta descends and starts to branch to the various organs. And as we'll see, the pressure will diminish as the branching continues. But the blood flows down pressure gradients from higher to lower pressures. I'm going to say that repeatedly. The only way blood flows in the body is if there is a pressure difference between 2 points. It could be a difference between the aorta and the carotid artery, the carotid arteries going up toward the head. It could be the difference between the aorta and the abdominal arteries or renal arteries. But the only way that blood will flow is if there exists a pressure difference. The pressure has to be higher upstream in order for the blood to flow downstream. It's really, at the end of the day, it's just that straightforward. Although we're going to look at it in a little more depth than that. This is a supplemental cartoon that I think you will find extremely useful shortly. It traces through that cardiovascular system in figure 14.1 in our text. It shows the pressure, relative pressure, starting in the left atrium, moving into the left ventricle, and then throughout all both the systemic and the pulmonary circuits back to the left atrium again. Notice the pressure is very low in the left atrium. It's almost zero. It's very low. But the ventricle, when it contracts, this is systole, diastole, systole, diastole. You get these large fluctuations in pressure within the left ventricle. Again, textbook values might be 120 over maybe 4 or 5 millimeters of mercury pressure. Blood moves into the aorta. Now again, the pressure, peak pressure is about the same as in the ventricle. I say about the same because when the heart contracts, it ejects the blood into the aorta. And the momentum when the blood is ejected into the aorta actually causes it to be just a touch higher than what's taking place in the ventricle. It's a fascinating thing, and we'll look at that in more depth later. But as the aorta branches, you see that basically the pressure pulses begin to diminish. And again, it's because the vessel is branching and giving rise to many, many smaller vessels. So the pressure is being spread out. Capillaries, you see basically no pulsation at all, nor in the veins, although there is actually a little bit. As the blood moves back into the right side of the heart, you can see that the right ventricle, again, low pressure, right atrium, very low pressure. When the right ventricle contracts, there's a greater pressure, but notice how much smaller it is than the left side of the heart. Again, it's a very useful diagram to compare the left and right sides of the heart. When I tell you that the pulmonary circulation is a very low pressure, low resistance system, you can come back to this figure and again, reinforce that concept by looking at this. So again, even though the left and the right ventricles contract almost simultaneously at the same time, pressures are much greater on the left side than the right side. I can think of at least 2 reasons for that. The muscle on the left side is much thicker than on the right side. But in addition to that, the resistance to the flow blood out of the heart is much greater on the left side. The blood's going to be ejected into the systemic circulation. The resistance is much greater there than it is leaving the right ventricle and going to the lungs. So those are reasons for that. So down below, I've given you some approximate diameters. I don't expect you to memorize these. The aorta, though, what I do want you to appreciate is that there's only a single aorta, and it's a large tube. The diameter changes somewhat. I'm giving it in terms of a radius here. A radius of 1.13, 2.54 is an inch. So the aorta, the internal diameter is about 1 inch. That's a big tube. It branches, and when you get to these smaller arteries, the radius of these might only be 0.5 millimeters. So there's many of these. We estimate, again, depending on where in the systemic circulation we look at these, but for smaller arteries, maybe 10 to the 4th. The arterioles, which give rise to capillaries, 10 to the 7th. Internal radius, maybe 15 microns, or actually less. Most of the arterioles Again, these are the resistance vessels. These are the ones which we're going to focus on a tremendous amount when we look at regulation of arterial blood pressure and the role that the autonomic nervous system plays in that. Capillaries, I can see here I've got continued branching. I've gone from a single tube, 10 to the 4th for the arteries, 10 to the 7th for the really small arteries, arterioles, 10 to the 10th for the capillaries. So I took the time to type this out because when you have a tube which branches into a greater number of tubes downstream, although the diameters decrease, the cross-sectional area, as we'll see shortly, increases. There's less resistance to the flow of blood downstream. And finally, the vena cava that returns to the heart, approximately the same internal diameter as in the aorta. So I'm recapping down here. At each branch point, the cross-sectional area of the daughter vessels exceeds that of the parent vessels. So in other words, the cross-sectional area of the arterioles exceeds that of the small arteries, which would be the parent vessels to the arterioles. Okay. I copied again, just pasted a couple of things on there which are useful. The mean arterial pressure in the aorta, maybe a textbook value, that's what that symbol there means, approximately about 95 millimeters of mercury. Comparing it with the pulmonary circuit, you can see that the mean arterial pressure is only about 15 millimeters of mercury pressure. That's astonishing, and you need to know that. There's a lot of clinical reasons for that. The pulmonary circulation, the blood is flowing through just tens of millions of capillaries. It's like a lake. The blood flow is slow. This allows a lot of time for blowing off the carbon dioxide, bringing in the oxygen so that the blood can be fully oxygenated before it's returned to the heart. The systemic circulation, I mean, let's think about that. We have to drive the blood from the heart up to the head. We got to drive it down to the abdomen, the intestines, the stomach. We got to drive the blood down to our legs, our toes. Teleologically, we need a pretty high driving force to achieve that. And again, we're going to see that's where a lot of the resistance resides, is in the systemic circulation. So again, I find this a very useful chart to refer to as I'm studying the cardiovascular system. So down below, I tried to summarize this for you. Total resistance in the systemic circulation is much higher than the pulmonary. Explains in part why the upstream driving pressure is so much greater in the systemic, 95 versus 15. There's more resistance to the flow of the blood. Okay. All right. We need to look at some basic hemodynamic principles now. Hemo refers to the blood, and hemodynamics, somebody coined this term. If you were going to take a course in hemodynamics, you would study blood flow rates, blood velocities, resistance to blood flow, and so forth and so on, viscosity of blood, and things of this nature. So the author has put together a collage of figures, and I'll break these down one at a time and go through them with you now. Okay, so what we want to do is to walk away from this discussion with an understanding of blood flow, what influences blood flow. Flow is mils per minute, mils per second, volume per unit of time. What will change blood flow rate? Well, if the heart beats faster, the blood flow rate should go up, all things being equal, right? We'd also like to have some understanding of the velocity of blood flow. That's different. That's how fast is the blood moving through the system. Okay, so we'll break each of these down. And what I've done is I've taken this figure, and then I've captured the first two images, and let me enlarge that. Again, I sometimes have to diminish the size on this so that I can read behind this stuff that's on my screen that you probably can't see. Let me see if I can move this. I can. See, even I can be trained. Okay. Can't move it any higher than that, though. Okay. All right. This figure, this is figure 14.3. This is part A and part B. The panel on the left, part A, you can imagine this as being a system of glass tubes filled with fluid. This could just be water in this case. They've got a cork in the end here so that they can prevent any outflow of blood. And the author here is simply trying to illustrate hydrostatic pressure. There's no flow going on here. The arrows indicate that the pressure is against the walls of whatever vessel that the fluid is in. There would also be pressure in these tubes extending up toward the surface like this. So this is truly hydrostatic, prefix, suffix. Hydro for water, hydrostatic. Stasis or stat means to stop. If I was doing surgery and somebody handed me a pair of hemostats, they would be like almost like a little pair of needle nose pliers that I could clamp off a blood vessel and stop blood flow. Hemostasis. Hydrostatic is just that. And as she points out, and she's correct, we tend to say, what's the hydrostatic pressure in the arteries? Well, the only way we can have hydrostatic pressure is if we stop blood flow. But when you read other textbooks and even current medical literature, you'll see the term used. You need to remind yourself they're really talking about hydraulic pressures. And even then, it's not water pressure, it's blood pressure, isn't it? So again, some of the nomenclature is off a little bit, but we can't avoid it. And that's why I'm talking about it with you now. You can read 2020 review articles on the cardiovascular system, and you'll see them use terms like that. So we just need to be aware of that and deal with that. So this would be hydrostatic pressure. If they pull the cork out and let the fluid start to flow, obviously the flow will leak out here downstream. The pressure that's being exerted here is the weight, the column of the fluid down on the horizontal tube and the fluid that's within it down below. Now, as the fluid drains off, you can see that the height of the water column diminishes. She's had the artist put arrows in here of diminishing boldness to indicate that the pressure would be less in this region than this region up here. And that's because of in this case, the hydrostatic pressure of the water down on the column of water that's in this horizontal tube. So it illustrates hydrostatic pressures. We have now a hydraulic pressure difference. The pressure is different upstream than it is downstream, and hence the fluid is flowing. Okay? Now, why does blood flow? Well, I've already given the answer away. I've said blood can't flow unless there's a pressure difference. We need to provide some sort of a semi-quantitative assessment of that. And we need to do that so that we can measure blood flow. We also need to understand what determines blood flow. One of the reasons that you're given equations to illustrate these phenomena is to show you what determines blood flow. It's not so much that we're going to be doing a lot of calculating of blood flow, but understand what determines it. And then we want to relate those determinants to physiological mechanisms. So to start out here, I've captured, first of all, the image from the text. This is a straight tube. In this text and almost every text you'll look at, they begin to look at blood flow through a system of straight, smooth tubes, as though they were glass, okay? They do that to simplify things. It's very difficult to make a model of an actual blood vessel, which is distensible, and which is not straight. They are not perfectly smooth inside. Wish they were, but they're not. And so we have to simplify that, and we do that by imagining a straight tube. So this shows a tube of some arbitrary length and diameter. We've got a pressure at this end and another pressure at the other end. Now, if blood is going to flow, there's got to be a difference between this end and the other end. In other words, P1 minus P2 at the other end has to be different. Delta just means difference. So there has to be a difference in pressure between this end and this end for fluid to flow. And in this diagram, she's just arbitrarily saying, I got a higher pressure here and a lower pressure downstream, so blood can flow. We can estimate flow rates using an equation called Ohm's law. This is when you take physics, you'll be exposed to Ohm's law with regard to electrical currents. But the same phenomenon, or an analogous phenomenon, can be assessed or analyzed using Ohm's law. In this case, F, or flow rate, equals the difference in pressure divided by the resistance. And what this says is that flow rate is directly proportional to the pressure difference and indirectly proportional to the resistance of blood flow or water flow through the tube, okay? So again, F represents the quantity of blood. It could be mils per minute. Down below, I've given other examples. For the whole system, we might talk about liters per minute. But any kind of volume per unit of time. Delta P, again, is the difference in pressure. And we most commonly see that given in millimeters of mercury pressure. And this text is no different. But delta P just means what's the difference between P1 and P2? R, the most difficult thing to measure, is resistance. So recapping, in the above equation, flow is directly proportional to the pressure difference, indirectly proportional to the resistance. An adult human male, flow rate is approximately 5 liters a minute. Okay? Again, we have about 5 liters of blood in our body. So about every minute, okay, we're pumping 5 liters. That's the flow rate collectively, a good ballpark estimate, okay? And it also equals the cardiac output. It's a closed system of tubes. It's a pump and a closed system of tubes. So it has to be the same. Interestingly, when you go working out, running can go up 5 times that. So instead of 5 liters a minute, your heart can be pumping out 25 liters a minute. What's the underlying physiological mechanism or mechanisms for that? That's the kind of thing we want to discuss in this course. Okay, flow rate. I wanted to show you this because it's another way to express flow rates, and also the cartoon you'll find extremely useful. Another expression, another way to describe flow rate is with the term conductance. And again, it's a measure of the volume of blood that flows per unit of time. That's similar to the flow rate that I just talked about, but for a given pressure difference. So in other words, we're normalizing flow rate. If the pressure difference between the left ventricle and on the left side of the heart and the right atrium on the right side of the heart is 100 millimeters of mercury pressure, if that's constant, if I assume it's constant for calculation, then how much blood is being conducted through the body? Well, the example I just gave is about 5 liters per minute, okay? So at a given pressure difference, okay, if I hold that constant, I want to show you how the flow rate will change if the diameter of the vessels changes. If it goes from an arbitrary value of 1, if we double it, or if we double it again. In this example, the diameter, an arbitrary value of 1 at a constant pressure difference of 100, we're saying that there's about 1 mil per minute of fluid flowing through this tube. If you double the diameter of this tube from 1 to 2, the flow doesn't double. It goes up 16-fold. If you take that tube and double it again, relative to the first situation, it goes up 256-fold. Now, the reason I find this really useful and important is that the vessels, for example, the arterioles, are going to change diameter under different physiological conditions, whether we're lying down, standing up, running, whether we have hardening or the arteries, okay? And as the diameter of the vessels change, the flow rate or conductance of blood flow through the tubes is going to change tremendously. So if I'm experiencing a vasoconstrictor type response in response to like epinephrine, the diameter of these tubes, many of them will diminish. So the blood flow will decrease. The pressure will go up tremendously though. Okay, that'll come a little bit later on here. So what does this all mean? I'm going to say this in about 3 or 4 different ways before I'm done, but as a first shot at this, the flow or conductance is proportional to the vessel diameter to the 4th power at any given pressure difference, at any given pressure difference. So in other words, if I double this to 2 times 2 is 4, 2 times 2 is 8, times 2 is 16. So if I've got an arterial at this diameter and it dilates, if it increases its diameter, if it doubles, flow through that tissue will go up 16-fold. So if that's skeletal muscle, for example, and we have local dilation of these vessels, the blood flow will go up 16-fold. Okay. All right. Now, don't panic. I'm only showing this cartoon for illustrative purposes. I don't expect you to memorize layers and all this other stuff. What I wanted to show you is that in an idealized tube, a straight tube, fluid, whether it's water or blood, tends to flow in a laminar type fashion. In other words, laminar just means layers, laminations, like you got a laminated floor. Okay. It flows in a parabolic fashion where the velocity is greatest in the center and least right at the interface between the wall of the tube and the fluid. One of the major reasons for that is that fluid flowing against the inner wall of a tube, like a glass tube or a blood vessel, there's going to be frictional resistance there. That's where it's going to be greatest. So the flow rate will be less there than the velocity will be less there than it would be out in the center. As you move away from the wall of the vessel, the water, like if this was water, the molecules are rolling across each other and there's less resistance, internal resistance in the fluid. And so the highest velocity would be out here. And again, if it was an idealized glass tube that was perfectly straight and smooth, you'd see parabolic flow like this. Again, blood vessels don't do this. They approach it in some cases, but they never make it there, okay? Something worth noting is that blood is more dense than water, about 5 times more viscous than water. And that's primarily because what are sometimes loosely recalled formed elements. That's red blood cells, white blood cells, proteins, things that have what molecules and structures like cells that when they bump into each other, they will exhibit some internal resistance to the movement of molecules and entities or elements. Okay, we can get a little more complicated with blood flow now, and we can integrate measurements from many different diameters of tubes and arrive at an expression called Poiseuille's law. Now, I'm going to describe this about 4 or 5 different ways, so don't panic, okay? In this first case, I'm looking at flow, and I'll show you a picture of him. I tried to put it on this image, but it wouldn't fit. He died in the late 1869, I think. He was right after the Civil War was when he died. At any rate, it was during the early 1800s that he worked out a lot of that knowledge that we have today. His relationships show that flow rate through a tube, like mils per minute, is directly proportional to, let me go through this slowly, pi times the pressure difference times the radius of the tube to the forced power. Now, again, I've already done that with Ohm's law, right? I said it's directly proportional to the pressure difference, okay? And inversely proportional to the resistance. So some of this is coming back at us again. I'm saying flow is directly proportional to pi times the pressure difference times the radius to the 4th power of the tube. It's indirectly proportional to 8 times eta, which represents the viscosity of the fluid, times the length of the tube. Again, he worked this out for straight, rigid cylinders, okay? Now, I've told you that when I give you equations like this, I'm not just going through a list of them, trying to give you some equations to memorize. It's to illustrate the effects that things like pressure differences, viscosity of fluids, length of tubes like blood vessels, what effect does that have on the flow of blood? Well, let me just pick one at random here. The length of the tube. Flow rate, if everything else was constant and all I varied was the length of the tube, let's say I tripled the length of the tube. Well, the denominator is going to get bigger. When I divide it into the numerator, this value is going to get smaller. So it says that as the length of the tube gets bigger and bigger and bigger, flow rate's going to diminish. Pick another one like viscosity. You can think of viscosity. I mean, everybody's seen a can of motor oil, and there's motor oil with different viscosities. Some are thicker, more viscous than others. As the viscosity increases, the same phenomenon occurs. This denominator gets bigger. When I divide it into whatever this number is, this number goes down. I've already told you that as the pressure difference increases, everything else held constant, as the difference in pressure gets bigger and bigger and bigger, the numerator is going to get bigger. So the flow rate's going to go up. So equations are useful, not really for the purpose of calculating precise flow rates, although they are used for that, but it's useful from a student point of view to teach ourselves the role that each of these parameters play in determining blood flow. And we're going to eventually be studying blood flow through the cardiovascular system. So I said, why are we taking the time to discuss all this? Well, I've given several reasons already, and I'm going to simplify this even more, but in fact, I'll kind of give you a clue now. The radius. How does the radius of the tubes change? Well, the radius of arteries and arterioles, they can change if there's increased sympathetic stimulation to them, or if you administer drugs, which have those kinds of effects. If it causes the smooth muscle around the tube to contract and constrict the diameter of the tube, the diameter will diminish. If that happens, flow rate, at least through that capillary bed, will go down. So these are the reasons why we're looking at this. And again, another cartoon which I found, which illustrated, at least in cross-section, these multiple layers, parabolic type flow that you would see in larger vessels that you don't see in smaller vessels. In fact, capillaries are of a diameter that, for most of the capillary beds, the red blood cells basically line up and go through one at a time. We used to illustrate this in physiology laboratories. A good model system is the frog foot. If you take a frog's foot and spread it out on a dissecting scope, you can actually see the blood cells move through the capillaries. It's like a roll of pennies that are moving through a tube. It's a fascinating thing to watch. Okay, so to recap, okay, flow is only going to occur when you have a pressure difference. So how important is the absolute pressure? Well, here's a tube with a pressure at the left of 175 at the distal end. So the delta P is 25. So you're going to get blood flow, right? Or water flow, or whatever's moving through the tube. Down below is another comparison where the tube it's much reduced by 60 millimeters of mercury pressure to 40 at this end and only 15 at the other end. The difference is still 25 though. And as it turns out, the flow is absolutely identical. So in this case, the absolute pressure doesn't matter. The flow rate will be constant. It's the pressure difference. This is a beautiful cartoon she's put together because it illustrates, again, blood is only going to flow, or fluid's only going to flow, if there's a pressure difference between 2 points. If it's just a section of a tube that I'm considering here, has to be a pressure difference for fluid to flow, okay? All right, now, here's Ohm's law again, the same cartoon which I showed you before. I want to point out that flow, again, equal to the pressure difference divided by the resistance. Well, now, even I can sit down and solve for each of these parameters. Delta P in this equation would be equal to flow times resistance. Resistance would be equal to delta P over flow. Now, resistance is one of the hardest things to measure. but we can estimate it and give us a sense of the level of resistance in a given portion of the cardiovascular system. And I've done that down below here, and I've said, okay, I'm going to use this R equals delta P over F, and I'm going to do it for the systemic circulation, the whole thing, and the pulmonary circulation, the entire thing. So here's a cartoon, okay? So here's the aorta, and we're just going to pretend we got one capillary bed here to represent the entire lower portion of the body, and blood's going to come back to the right atrium. And so we're going to say, what is the pressure difference, the delta P, between the, for example, the left ventricle and the right atrium? Textbook value about 100. Now remember, a mean arterial pressure, let's say 95. And I showed you in the graph that the pressure in the right atrium might be almost 0. So it's about 100. Okay? Flow rate, remember, 5 mils per minute. Let's say about 100 mils per second. If you divide these numbers out, you get something called a peripheral resistance unit. And of course, 100 into 100 is 1. We did it intentionally just to make it really just dirt easy. Okay? If you compare that with the pulmonary circulation, okay, the resistance in this case, the difference in pressure between the blood being pumped into the pulmonary arteries and returning to the left atrium, it's only about 14 millimeters of mercury pressure difference. And down below, I've indicated, and I'm just using values of about 16 arterial, 2 venous. But because the system is a closed system of tubes, the same volume of blood must move per unit of time. It's a closed system. So 14 divided by 100 gives me 0.14 peripheral resistance units. You can see that estimating, and we are estimating, the resistance in the pulmonary circulation is about 1/10 of what it is in the systemic circulation. Much, much less. Low pressure, low resistance. High pressure, much higher resistance in the systemic circulation. All right. We're nearing the end of today's discussion. I'm going to use Poiseuille's relationship to now look at resistance. And there's a photo of the man. Yeah, he died. Yeah, I was right, 1869. Based on work that he carried out, as you can see, in the early 1800s, you can estimate resistance. It's equal to 8 times the length of the tube times the viscosity of the fluid divided by pi times r to the 4th. In other words, it's directly proportional to length and viscosity, indirectly proportional to the diameter of the tube. Now, lots of these equations, we can simplify them because there's constants. The 8 and the pi, okay, they can drop out, and we can say, well, resistance is proportional to length, viscosity divided by diameter, or radius to the 4th. Well, it turns out viscosity physiologically doesn't change very much, and neither does the length of the tube. I mean, arteries and veins don't really change length in our bodies. And the viscosity of the fluid, at least physiologically, changes very little. Now, there's differences in viscosity of blood in different regions of the body, and I'm not going to get into that. That's way beyond the scope of this course. But in general, the viscosity doesn't change. There's pathological conditions, polycythemia vera, where viscosity increases and so forth. But this is not pathology. This is just straight human physiology. Viscosity does not change very much. Neither does the length of the tubes. So I can really simplify it to resistance is proportional to, I'm estimating it, that's why I don't have an equal sign there. Resistance is proportional to 1 over the diameter, or r to the 4th, radius to the 4th power of the tube. So again, summarizing this, resistance increases as tube length increases, resistance increases as viscosity increases. Neither of those turn out to be really important. Resistance decreases as diameter increases. That's real important. In fact, before I put this together for you, I change the color on some of these things to draw your attention to this. I think it should be apparent why we study the underlying physiological mechanisms that regulate vasoconstriction and vasodilation of blood vessels. As the diameter of the blood vessels change, whether it's an artery, an arterial, a vein, it has a profound effect on the level of resistance to the flow of blood through those tubes. Hence, blood flow changes. Blood flow, as we'll see, is regulated on a local basis, not only on an organ-to-organ basis, but even within local regions of tissue beds. Lungs are a beautiful example of that. Blood flow is not constant throughout all of the lung arterioles and capillaries. And I'll give you some exquisite examples of that when we get to the respiratory system. Okay? All right. One last thing to discuss, and that's the velocity of blood flow. Again, the flow rate is volume per unit of time. Velocity would be just like a car going down the racetrack or me trying to run a race if I could. It's distance per unit of time. So it's centimeters per minute, or this is just one way that I could express it. In other words, a simple way to say it is how fast is the blood moving? All right. In this example, we have a tank and we have outlet tubes. An arbitrary radius of 1, you can make it inches or centimeters or whatever you want. And over here, we've got a radius of 2, okay? So if you look at the resistance, and again, we've already established it's proportional to 1 over the diameter, or r to the 4th to be correct. Tube A, okay, radius of 1, it doubles in over here on tube B to a radius of 2. Resistance is proportional to 1 over the radius. So 1 over 1 to the 4th is 1. 1 times 1 is 1 times 1 is 1, and so forth. So the resistance, arbitrary resistance unit in this case, is 1. Tube B, because this is now 2, the radius is 2, it's 2 to the 4th. 2 times 2 is 4, times 2 is 8, times 2 is 16. So the resistance, okay, is different. It's only 1/16 of what it is over here, okay? Because the denominator increases in size, this value becomes less mathematically. So the resistance diminishes. And again, she equates that with flow, okay? And I've already done that with flow. It's basically the same thing. So we're equating the resistance to flow, okay, based on radius. And here we actually look at the actual flow rates. So this is flow rate. This is the resistance to flow. Now, what's that have to do with velocity of blood flow? Conductance, C, again, 1 over the radius. Oh, I'm sorry, 1 over resistance, okay? Flow rate and velocity are different parameters. Velocity of flow, again, is distance. A fixed volume of blood flows through a given period of time. So centimeters per second, centimeters per minute, any of those arbitrary units, I could utilize that. Velocity can be equated to flow over cross-sectional area of the tubular system. It's a pretty simple relationship, really. So again, V, little v is velocity. I'll just say centimeters per minute. Q in this case is flow rate, volume per time. In this case, we would make like cubic centimeters per minute. And then A is just cross-sectional area. Okay, now, 1 mil of blood is about 1 cubic centimeter. In the cartoon bound below, she's got fluid flowing through a tube of 2 different diameters. She wants to look at the effects of cross-sectional area on the velocity of blood flow. That's why the cartoon is set up this way. The flow rate in this case, this is it's at a given value or constant flow rate. So flow rate in this case, she's got fluid coming in at the left at 12 cubic centimeters per minute. Let's say 12 mils per minute, okay? If the blood comes in at 12 mils per minute, will the velocity be constant as it moves through a tubular system of varying diameters? Well, the answer is no. It's a trick question. If you calculate the velocity of blood flow in the narrow diameter of this tubular system, okay, the area here is a square centimeter. So she's giving me the A value, she's giving me the Q value. So she puts it in a table form over here for me. She says, Jack, at this point X, velocity is equal to Q over the cross-sectional area, flow rate over the cross-sectional area. The flow rate is 12 cubic centimeters per minute, 12 mils per minute, divided by a cross-sectional area of 1 square centimeter. 1 into 12 is 12. So I've got 12 centimeters per minute velocity. We move out into the larger diameter, and she says we're going to increase it 12 times now. 12 times. So again, the flow rate's coming in. She hasn't changed that. She's changed, the only variable here is the cross-sectional area. So she tabulates that for me. She says, Jack, at point Y, velocity equals Q over A. Q is constant, I'm not going to vary that, but I'm going to increase the area to 12 square centimeters. Well, 12 into 12 is 1. So the velocity decreases 12 times because the cross-sectional area has increased 12 times. So flow rate can remain constant, but the velocity can slow down. Now, again, what do you care about that? Well, the blood flowing out of the pulmonary trunk, okay, that's leaving the heart. We got about 5 liters per minute. The blood flows through the lungs, though, millions and millions and millions of capillaries. The cross-sectional area goes up. So an analogy might be, here's the pulmonary trunk, here's the pulmonary cross-sectional area of capillaries here. Now, it's much greater than this, but I'm just using this as an analogy, okay? So even though the flow is going to stay constant, the velocity is much less. Why should I care about that? Well, it gives time for the blood to get rid of the carbon dioxide and pick up the oxygen. Similar analogies will exist for all the capillary beds in the body. The flow rate, the velocity is going to slow. Teleologically, I'm assigning a benefit to that. I'm saying that that allows an optimal amount of time for nutrient and waste product exchange. So these are the underlying physical principles for nutrient and waste exchange. Okay. A simple cartoon to pull this together for you. This again shows a typical artery, arterioles, capillaries, venules, veins, and eventually, a vena cava going back to the heart. So down at the bottom, you can see, again, a big artery like the aorta, arteries, arterioles, capillaries, venules, veins, vena cava. If you look at the total cross-sectional area, again, there's only one aorta, okay? What happens, though, as you branch into the arteries, smaller and smaller arteries, the arterioles, the capillaries, the cross-sectional area goes up tremendously. Look what happens to blood flow. I'm sorry, velocity of blood flow. Flow has to stay constant, right? overall. But the velocity slows tremendously. Again, overall, the flow has to stay the same. If the aorta is putting out 5 liters per minute, it's a system of closed tubes, 5 liters per minute has to be coming back, okay? But the velocity can change tremendously. And that allows for different levels of exchange of nutrients and waste. We decide that that's a physiological or a biological advantage, okay? All right, one last thing. I said that the heart provides this driving pressure, this mean arterial pressure, okay? Using a similar cartoon, this shows blood pressure starting in the aorta. Here's those systolic, diastolic pressure waves. They're diminishing, okay, as the vessels branch. And again, come back into the vena cava at almost 0 at the other side, okay? So systolic, diastolic, I'll dissect all of that out for you in detail later. Here's this mean pressure though. It's not an exact average, as you can see. It's not the middle of this. It's a little bit different, and we'll talk about that later. But mean arterial pressure is proportional to, we sometimes say equal, and I've cheated over here and done that, but it's proportional to cardiac output, liters per minute, mils per second, whatever, times peripheral resistance. And I've pasted this all over this to just draw your attention to this and indicate that you probably want to understand that. Mean arterial pressure equals or is proportional to cardiac output times peripheral resistance. We will look at regulation of cardiac output. We will look at the physiological regulation of peripheral resistance. If peripheral resistance goes up tremendously, that's a back pressure on the heart. So mean arterial pressure will tend to go up. Simple example here. Let's say that I'm given a drug which is a vasoconstrictor, causes my arterioles to constrict, millions of them to constrict. At that instant, if that's the only thing that happens, okay, cardiac output times peripheral resistance, the mean arterial pressure is going to go up. Okay, my blood pressure will increase. And indeed, patients are given vasoconstrictors to increase blood pressure. Okay, systemic regulation of blood pressure can result in increased blood flow to the brain and other organs of the body. So that can be obviously of clinical significance. Okay, all right, I'm going to stop now. That's taken me a little over an hour to do that. The next lecture, lecture 2, I will begin to discuss the underlying basis of the cardiac rhythm. In other words, what establishes sinus rhythm and how is it regulated physiologically? So I'll stop at this point. I'll have this posted on Moodle for you. And I think what I'm going to do is start labeling these. I'll label this one Lecture 1, Chapter 14, Part A. The next one will be Lecture 2, Chapter 14, Part B, and however many sections we need to get through in order to finish that. So I'll stop at this point and have this posted for you. Have a great day, and I'll be coming back to you soon.